Good morning, everybody. My name is Zhang, and I'm the Deputy Manager at Flow Cytometry Facility here at LMB. Welcome to the Biophysical Technique lectures. So this morning, I'm going to give you an introduction to flow cytometry. So I'm going to talk through our flow cytometry facility, who we are and what we do. What is flow cytometry, how a flow cytometer works, followed by sample preparation, and most interestingly, probably useful, to show you applications we have done in our facility. So there are three of us, and Pierre-Andre Pantila is the head of facility, and myself and Yichen. We locate at 3N185 and 3N180. 3N185 is our main lab. You are welcome to come to talk to us anytime. So there are two main categories of flow cytometers. One category is cell analyzers. We have 6 cell analyzers in our lab. 4 are conventional cell analyzers. As shown in here, we have 2BD as alpha Tessa and LSR2 and back computer Cytoflex XL. If you go to the internal website here, and under the fluorophore in our facility webpage, and you will find all the spec for each instrument, which could be quite useful when you design your panel. We have 2 brand new Sony ID7000 spectral analyzer. Very exciting, they are brand new kits. And they equipped with 6 lasers, they having 184 PMT detectors. They are especially useful for high dimensional analysis for the big panels for immunologists. We have 4 cell sorters. And from the left is this older module and synergy. And in the middle is BD fax area fusion and 2 Bigfoot spectral cell sorter. They are all high-speed sorters, cell sorters. Again, you can find the spec under our webpage. Or just come to talk to us what you want to do. We can point to you which one we're going to use. To book time on our cytometers, again, you're welcome to talk to us, come to talk to us. To book sorting, email to CellSort email address and tell us what you want to do, what cell type, and how many cells you want to sort. We can book, we can agree time, book on our calendar. To book analyzer, you come to us for training. After the training, you can book yourself on the Carrier Connect for each instrument. The cell sorting is available from during the working hour because we perform the cell sorting. And to analyze your cells, and you can book the instrument anytime, 24 hours, 365 days, as long as you book yourself on this calendar. So there are other types of cytometers. We don't have it in the lab. However, we have access to them across the CRUK flow cytometry facilities. If you come across them and you want to use them, please come to talk to us. We can facilitate the usage. So there are image streams, so you can capture an image while you're analyzing the cells. However, the comes with pros and cons. Come to talk to us. It'll be easier to understand how you use these kind of flow cytometer. And also there are mass cytometry and image mass cytometry. Again, we can get access to them in CRUK. Mass cytometer use a metal tag rather than fluorescent tag on antibodies. It's particularly useful for the large panel. For example, the cancer patient sample, you have very limited amount of sample. And the metal tag reduce the spectral overlap. So make analyzing the sample easier However, also it comes with disadvantage. For example, it's very slow. So come to the concept, what is flow cytometry? It is a laser-based technology used to analyze the characteristics of cells or particles in the fluidic system as they pass one by one through a sensing point. It measures fluorescent intensity produced by fluorescent labeled antibody, detect protein or ligands that binds to DNA. So flow cytometry can analyze the expression of cell surface marker or intracellular molecules. It characterizes and defines different cell population. It can analyze cell size or do cell count. It allows simultaneously multi-parameter analysis of single cells. So flow cytometry compared to the microscope, it comes with its own advantage. So, but flow cytometer cannot tell you where the antigen is. However, it can analyze many cells in a very short time frame. Also look at numerous parameters at once. So before we go on and dissect a flow cytometer, I like to mention fluorophores and fluorescence again. If you use a microscope, you're probably very familiar with fluorophores and fluorescence. Fluorescence is the emission light is the emission of light by certain molecules, which are fluorophores. The fluorophores or fluorescent molecules absorb external energy, the electrons raised to the excited state, however unstable. When it returns to the ground state, the molecule emits a light. Every fluorophore comes with its own absorption spectrum and emission spectrum. Knowing its absorption spectrum and emission spectrum will help you to design your panel. So all these fluorescent molecules, fluorophores, or we call it dyes, they come with single dyes or tandem dyes. The single dyes, for example, fixate PEA, PC, they just fluoresce on its own. And the tandem dyes, they are derived from single dyes, which uses the FRET. The donor accepts the excited by external energy. The emission lights overlap with excitation light of acceptor molecules. Okay, shown here. And the acceptor molecules emits a longer wavelength of the light. So I have some tables showing the tandem dyes we commonly used in our panel. So why we use tandem dyes here? The main purpose is to increase number of parameters that can be detected at the same time. And for example, take the BD Horizon Brilliant violet dye as an example. The first BV421, we can see, to BV510, they are single dyes. All these dyes from yellow and red range are derived from these single dyes. You can see all these dyes occupies almost all the visible spectrums can be excited by a single laser line. The same to the BD Horizon Brilliant UV dyes. So we have so many dyes, so many options, and we can pick and combine different dyes together to design our panel. So we also have fluorescent proteins. It originally come from natural proteins, for example, GFP from jellyfish. And then these fluorescent proteins further validated and mutate to a stable molecules. For example, we have GFP, CFP, YFP. You probably already don't know this, not only used for flow cytometry, maybe used for your imaging. So these fluorescent proteins can be encoded with your protein of interest, acting as intracellular markers in live cells. So let's talk about the components of flow cytometers. Let's see how it works. So first part, let's have a look at fluidics. A fluidic system of a flow cytometer is to transport cells or particles in a stream to the interrogation point of the laser. Shows in here. So this fluidic system or fluidic stream should be free of air bubbles, dirt and clumps, should be very stable. Ideally, the cells or particles are transported in the center of the laser beam. Only one cell passing through this sensing point at one time. So flow cytometer use the process of hydrodynamic focusing, where the sheath fluid pressurized and flow through the flow cell or a nozzle, as shown in here, at a constant rate. And sample stream, or we call it a sample fluidic system, sample core stream is injected in the center of the sheath fluid. So this sheath fluid force the sample core passing through the point of the sensing point in a single file, which means single cells or particles passing through the laser beam at a given moment. So we measure the cells one by one when they're passing through the laser point, sensing point here. Although the sheath fluid is constant, we can change the sample flow rate to increase our speed of acquisition. However, to increase flow rate, we have to open the core stream here. It comes with disadvantage. If you want to running your samples much faster, you have to allow more sample core stream passing through the lasers. More like larger amount of fluid, the core stream passing through the sensing point. You can see the cells no longer going through the middle of the laser beam, they are deviates. So it can decrease the resolution and increase coincidence. However, there is a time strain as always. So there's a balance between flow rate and the time. So we should have looked the optics. In our flow cytometers, we have a good range of excitation source, which are lasers. We have so many, we have a range of the lasers at different wavelengths. These laser lines are quite sufficient to cover the needs of the most fluorophores on the market. So these lasers are shaped and focused onto the sample stream by a series of lenses. So I have to mention the optical filters, which capture the incoming emission light. There are two types of filters. One is called dichroic mirror, which is splitting the incoming light, and the other part is bandpass filter. So take an example. So there's an incoming emission light. If it encounters 540 nanometer dichroic short pass mirrors, any light shorter than 540 pass through, and longer than 540 nanometer will deflect at a right angle, as shown here, to enter the next optical path. So this light are further collected by the bandpass filter. We can have look later in a diagram, probably will show you more clearly. So let's summarize what we talked about previously onto this simple diagram. So when your cells, your samples injected into a nozzle or fluidic system, they are being forced into a single file. One cell passing through this sensing point at any one time. The emission light from probably multiple fluoroforms. So the emission lights from multiple fluoroforms are collected by this lens and enter the optical path. So, for example, the emission light encounters first dichroic mirror. Anything longer than 510 pass through, and anything shorter than 510 gets deflected and collected by this bandpass filter and enter the detector. So the longer than 510 will go through and encounter the next dichroic long pass filter, so on and so forth. By this way, the multiple fluoroforms can be detected if they exist on the same cell population. So apart from conventional cytometer, we are at an exciting time, exciting time of next generation of flow cytometers, spectral technology. So we have 2 spectral analyzers in the lab. So our immunologists already moved on to use the spectral analyzer. They basically can fit very large panels. So the main difference between the spectral and the conventional cytometers is the optical path and how they collect the light. So rather than collect only a band light of the emission, and spectral analyzers utilize this light diffraction grating technology. Basically, they are distribute incoming emission light and diffract them, the whole spectral wavelength onto the detector, which means the whole emission spectrum are collected. So the software can further deconvolute the combined light by an algorithm. Here we use weighted least square method. They can deconvolute the colors that in the combined emission light by comparing the combined light to the reference, single color reference spectrum. If you are interested to doing larger panel, or you're an immunologist, please come to talk to us. Here, I'm only giving you very simple introduction of spectral technology. It's more than what I'm saying here. So why we use spectral analyzer? So the most advantage is to separate highly overlapped dyes. For example, APC and Fluor 647, before we cannot use them in the same panel in the conventional cytometer. Now we can fit these highly overlapped fluoroforms into the same panel. So basically, this allows us to expand the panel size significantly. This is particularly useful when there's very limited amount of sample available. Also, another advantage from the spectral technology is autofluorescence subtraction. So I give an example here. This is the sliced whole blood samples, and we can see on unstained sample, we already have this background coming from various populations from this blood sample, and any signal from BUV 395 will be masked. By perform autofluorescent subtraction here, we clean up the plots, and we can now review any weak signal coming from the BUV 395. So this is a very powerful aspect of spectral technology. Even you only want to have a look the autofluorescent profile of your samples, you can use spectral technology. You can have some idea about your cells, how they do, where they fluoresce. So they'll be really useful when you come design your panels. So electronics part of flow cytometer, basically the detectors will convert incoming photons to electric currents. There are two types of detectors on our flow cytometer. One is PMT, is APD. You won't see this, but depends how the manufacturer designed flow cytometer. They are doing the similar thing. So the electronics can analyze pulse, height, width, area, and digitalize the signal and store the data into the computer. So this is how the pulse, voltage pulse measures. When your cells enter the laser and the pulse rise and reach the peak when the cell is fully immersed in the laser beam and return to the ground, return to the baseline once the cell exits the laser beam. The whole area, width, and height are measured. So how we collect the data, how we analyze the data on a flow cytometer. So I have to introduce force scatter and set scatter. Putting the fluorescent signal aside, the flow cytometer gives you two free parameters, which is force scatter and set scatter. When cells enter a given laser beam, the cells itself diffract light in all directions. We have a detector placed at forward direction, collecting the light basically facing the laser beam. So if your cells are uniform, you see the larger cells will deflect the light at a larger angle. So if your cells are uniform, the force scatter will be measured. The measurement from force scatter is related to your cell size. However, if your cells are not uniform, that you have to be careful because that might not directly related to can measure your cell size. So the force scatter give you idea of the cell size, but they are not the measured parameter for your cell size. So basically, you cannot report your cells are bigger or smaller, just only by looking at force scatter. So set scatter, similarly, the set scatter collect the light at 90-degree angles. Set scatter is related to the complexity of the cells. So for example, macrophages will deflect the light at a larger angle. So you can you can see the different populations by only looking at the set scatter already here. If you combine the two parameters together and plot them onto the two parameter dot plot, you already see you can see the three distinct populations here already. So this is example to show you how the force scatter do in the whole black dot plot. So these three populations indicates lymphocytes, monocytes, and neutrophils. And of course, we have a thresholding on force scatter signal to cut off any unwanted signal. So similarly, the fluorescent signal coming from the fluorophores that are present on your cell surface or internally. So the fluorescent intensity is related to the abundance of your protein on the surface or inside the cells. And you can see the voltage pulse will become larger when the protein density are greater than the least abundant protein. So these are the fluorescent dot plots. Let's take an example again. We can see the three populations in the fresh whole blood. And if we have fluorescent parameters, we can plot into a 2D plots, and we place a gate and a region around the cell of the interest. And we can gate this region to the next plot to further analyze the subpopulation. All flow cytometry data comes and stored as FCS files. So these FCS raw files can be read by the third-party analysis software, FlowJo and FCS Express 7. We have site license of FlowJo. If you want to analyze flow cytometry data, please contact me. I can give you a license. There are some statistic tools to help you to calculate percentage of your positive cells or compare the intensity of fluorescent signal, or to see, especially for when you're measuring the DNA, the CVs are important parameter to look at. So we not only can analyze the cells, we can also separate a cell population of the interest from your sample. I'm not going to go into the cell sorting a lot. On Thursday, the head of facility, Pierre-Andre, will give you another talk focusing on cell sorting. If you're interested, please attend Thursday's lecture. So basically, if you stain your cells with different fluoroforms and apply them onto a cell sorter, so this in the population of interest can be sorted electrostatically and collect them onto the collection tube. This population can be subject to a further analysis. So briefly, this is how a cell sorter looks like. In upper chamber, that's where you analyze your samples. You see there's an interrogation points with different laser lines. After that, and all the string will be shaped and generate many jobs. Your cells will be wrapped into these jobs. And when the cell of interest travel to the last detached job, that's when the whole stream is charged. And this job will be deflected and collected into a medium, for example, tubes or plates. So we can sort your cells into tubes, 5 ml tubes, 1.5 micron doors, plates, many different sizes of the tubes, I would say. Sample preparation is the key to have quantitative data. So some cells are easy, come naturally in suspension. If your cells attach to a culture plate, please be careful when you dissociate them. And basically, we want single cell suspension. The first principle of flow cytometry is rubbish in, rubbish out. There's no magic. So we have, here shows you a cell strainer. If your cells are clumpy or before you apply them to flow cytometer, please filter them through this nylon mesh. So they come with different different size of pores, 50 micron or 70 micron. For cell sorter, we have 70 micron nozzle, 100 micron nozzle. So, you know, 2 cells stuck together will block our machines, and we have to start QC again. So other considerations, please dilute your sample for a concentration the instrument can handle. Again, you can talk to us about this point. So get rid of any unwanted cells, for example, red blood cells, because every event counts. So that will slow down your acquisition. Or if you are preparing the tissues or blood samples, you can get rid of something like granulocytes if you're only interested in lymphocytes. Including a viability dye is the key to get out the dead cells. There are many viability dyes on the market you can choose to fit into your panel. So just only a simple example to show you, without a viability dye, you can see this false population. Looks like a double positive from Kirla G1 and ICOS. Looks like there's a weaker LC2 population. However, this population coming from the dead cells. So after getting out the dead cells using a lab dead marker, you can see there's much cleaner population. Always including controls. So the people always ask me, if I need burden controls, yes, because there's variations between the days, between experiments, between the samples. So for each experiment, at least you need negative controls. If you do multicolor experiment, please burn your single color controls. So the positive population that detected on the cytometers are always related to your negative population. The basic principles is brightest dye on least expressed energy, and choose the dyes with least spectral overlap. So come to talk about flow cytometry, we cannot avoid spectral overlap. These are examples. The spectral overlap means unwanted signals spill into the channel that we are detecting here. So I take some examples here. So if we are running single color controls, we can assess the spectral overlap. For example, if we're running alexafluor-488, we know it's only alexafluor-488. However, if we plot the 488 against the PE, we can see the signal from 488 spilling into the PE channels. So we will need applied compensation calculation to correct the mistakes. So single color controls are necessary for multi-color experiments. So this is an example if we don't do color compensation, and you can see this fixes spill into the PE channel, this population looks like double positive. After applied compensation calculation, we can see the fixate falling into its own place, PE as well, and there's no double positive population as supposed to be. So we are coming to show you some applications examples we have done in our facility. We always open to new application. If you have new ideas, we always wanting to add in your application into our portfolio. So the simplest one we always do is fluorescent proteins. Now people doing CRISPR knockout using a fluorescent protein markers before they're using plasmid. You can see you can use fluorescent protein markers. However, the expression of your protein related to the fluorescent, the protein of your interest that tags to your fluorescent protein, the expression pattern can be different. So this is a negative population. Here you have a nice positive population distinct from negatives. However, in some of the cells, they actually has continuous expression. So again, your controls are important. Without controls, it's hard to place the gate where the population interest should be. So not only these two fluorescent proteins, we detect all range of fluorescent proteins, IRP and CHARI depends. You also can use a combination of fluorescent proteins into in your experiment. This is confetti mice generated in Mackenzie's lab. So they have this fluorescent protein coupled with transcription factor, Katusha, TD tomato, EFP, and TFP. So before you design your more complex panel, please come to talk to us so we can check if all these fluoroforms or fluorescent proteins are, can be detected on our flow cytometer at the same time. If they are highly overlapped fluorescent proteins, will make your analysis harder. So we can also look at the apoptosis. So as you know, when the cell undergo apoptosis, the protein phosphatidylserine that exists in the internal of the cell membrane, and when it undergoes apoptosis, the protein will flip to external membrane of the cells. We can detect by annexin 5. We usually combine annexin 5 with probably an iodide. So in this plot, you already can see there's a single positive annexin 5 indicating apoptotic cells. And when the cells are at later age of apoptosis, the cell membrane are compromised, so the PI can enter the cell membrane state, stain the DNA. So here shows the dead cells, which are double positive from PI and 5. So we can also track the proliferation using CFSE or other cell tracing dyes. So here is an example. We load CFSE to the parent cell population. When the cell divides, the fluorescence halves. So they gone through several generations of division, and the fluorescence is becoming weaker and weaker. So we can track this by plotting the fluorescence here, histogram, plotting this fluorescent dye here, so we can see the parent and the fluorescence, we're losing fluorescence across the division cycles. So here's example, the T cell proliferation from mouse spleen. First the gate, of course, we select our cells and single cells. And here the cells was loaded with cell trace violet. We can see the cells undergo several divisions. These are the T cells. When they divide, they express L13. So their L13 act with the GFP, fluorescent protein. We can also analyze a cell cycle by flow cytometer. There are a number of dyes available on the market. So typically people use Hirsch and DAPI, sometimes use a PI. So when you do cell cycle analysis, please come to talk to us because there are certain settings has to be set on our flow cytometers. We are detecting double amount of DNA in the G2 stage compared to G1. So there's very subtle difference between these two stages. So as you can imagine, if the two G1 cells enter the laser beam, it can be detected as one G2 cell. So to gate out the clumps and the doublets is essential in cell cycle experiment. So here's a human myeloid leukemia cell lines. They having haploids and diploids, and we can see the diploids go through the cell cycle, G0, G1S, and the G2. So when you do your cell cycle, you load the DNA dyes. So please be aware, titrate. You need to titrate your dyes into appropriate concentration. More dyes doesn't mean you have a better resolution. For example, here, so the best resolution achieved at 312 nanogram rather than 20 microgram. Actually, you at this high concentration, you actually lost complete resolution. And from so this is data from preschool. These are showing our clear cell cycle analysis combined with the division markers. And these archaea cells has very small genome, but they still be able to detect by flow cytometer. As you can see, we gated on the single cells, and these cells are loaded with Hirsch because Hirsch stain the live cells. Now we can see the cell cycle. When we plot the cell cycle against surface marker, we can see the expression patterns across the cell cycle here. So another thing I want to mention is when you analyze the cell cycle, rather than use log as an immunofluorescent, like most of fluorophore, you should plot them on a log scale. On a cell cycle, and you want to plot your cell cycle on a linear scale. So in the cell cycle, we can pick out S phase particularly. So Guiyang from Sales Lab gave a very good examples how to analyze the S phase within a cell cycle. So they use BRDU. So BRDU is a thymidine analog, can be incubated with the cells. The BRDU is incorporated into the genome. Later on, the BRDU can be detected by anti-BRDU antibody. So in this paper, and Zhang showed, compared to the wild type, we showed beautiful cell cycle S phase in polyploid MEF cells. These MEF cells has abnormal genome size. They are lost ability to divide. You can see they are actually accumulating the DNA rather than dividing. We can also do chromosome analysis in LMB. So our cell sorters are equipped with specific laser lines, 445 nanometer laser lines. These laser lines particularly is to look at chromycin A3. So chromycin A3 binds preferably to the GC content. DAPI preferably binds to AT content. So when we stain with DAPI and chromycin A3 into our chromosome, we can pick out each individual chromosomes. So we start doing chromosomes from a couple of years ago. And Sanger, apart from Sanger, there's, as far as I know, is only us in doing chromosome analysis. We particularly purchased a 445 nanometer laser line to do a chromosome analysis. So we can also use aquadine orange to assessing DNA integrity. And Anna Webb from PNAC did this beautiful experiment to assessing DNA integrity in sperm cells. So the aquadine orange intercalates into double-stranded DNA, emits green, and it stacks on single-stranded DNA, emits red. So when there's a procedure, there's a protocol called sperm chromatin structure assays. If you're interested, you can look up on this. Basically here, so here it shows you normal sperm cells with double-stranded DNA. Here is apotic and dead sperms. And more to the right, more DNA fragments are seen. And on this region, it's showing abnormal amount of the histone in the sperm cells. And it can be plotted as high DNA stability index. So the Hannah did this experiment across different mouse lines. The wild-type mouse lines and double knockout PRIM2 genes, and also the mutant PRIM2 genes. So we can see here, the green signal shifted to the red region significantly. Also, we have this abnormal amount of histone in the sperm chromatin here. So also on the PRIM2 mutant, we're seeing a similar thing. So all this parameter can be reported as a DNA fragmentation index or high DNA stability index, and plots in here and work out the statistics. If you're interested to doing this kind of experiment, it's a little bit tricky. You have to swap some of the filters on our cytometers. You come to talk to us, we can set up for you. So best friend of flow cytometry is immunophenotyping, of course. So these are the simple staining protocols. So again, I have to emphasize the importance of titrating your antibodies. You will want to use appropriate concentrations, ideally just the concentration enter the plateau. Then in this way, you achieve the best resolution with reasonable amount of the antibodies. The antibodies are quite expensive. So these are the simple examples for immunophenotyping in fresh whole blood, and they can phenotype different cell types already from fluorescent scatter and CD45 for the white cell. And subsequently, they're analyzing different types of T cells and B cells, myeloid cells, and also the activation stage of these cells. So these experiments is to characterize the hemopoietic stem cells. So they are used 3 different strategies to identify HSC cells with conical markers, with slab markers, and also with set populations. These stem cells, interestingly, pumping out the host. So you can see that this little tail that looks like a very least amount of the host and distinguished from the main population. So this is always my good examples to show you how to characterize HSC cells. So these HSC cells can be isolated by a cell sorter. So Huang and Jerry, years ago, they did this experiment. They isolated HSC cells was quite difficult because the frequency in the whole in their sample, sometimes 1 in a million, sometimes it can be. So we isolated the single cells from the host mice and transferred this cell back to the acceptor mice. And they are studying the reconstitute of the HSC cells in this host mice in different tissue. So this is how they did wide genome sequencing when they transferred the HSC cells into the host cells across different mouse lines. They are studying the DNA damage, as in here in they studied the DNA damage in alcohol endogenous aldehyde damage chromosomes. So we can looking at the surface markers. We can also look at intracellular cytokine staining. So when you're looking at the intracellular cytokine staining, you have to fix and permeabilize your cells, so the antibody can get access to the access into your cells. And here's examples. So there's this experiment is to study the cytokine production within T cells, and especially under the T cell division. So we can see here. So we can see here when the cell undergo the cell division, we can see the cytokine production in the cells. Our C2s are produced when the cells are dividing and not interferon gamma. We can also look at transcription factor in the cell population. But difference is when you want to look at transcription factor, the staining process will be more vigorous because the dyes need to enter the nucleus. So here, Noi gave us a good examples. He studied all the transcription factor, GATA3, rho gamma T, UMIS in the NK cells. So what I want to mention is you have to be careful about the staining process. There are protocols and also the transcription factor staining buffer available on the market to help you to stain these cells better. So these data is coming from Noi, and he analyzed his a sample on the spectral analyzers. It's very hard to, when he used over 30 color panels, it's very difficult to plot, 2D plot one after the other to locate where your cell population of interest are. So he uses this high dimensional analysis. If you are into computational biology, you're probably familiar with all this algorithm, UMAP or Tisney, SPADE. Basically, these clusters are showing the cell population having the same phenotype. So from the cluster, you can you can get further to analyze what actually markers expressed in those clusters. So this high dimensional analysis particularly useful for to discover the unknown population of the cells. So here Noi identified the ACE and KP cells, which act as a progenitor for LC1 NK cell population. Flow cytometer also can measure the calcium influx. Usually we use the dye called Indo-1, which is rheometric dye. So the dye, when it bonds to calcium, emits at 405 nanometer. When unbound, emits at 475 nanometer. So you can use the ratio to measure the calcium influx. Here, Aiden did calcium influx assays on CD4 positive naive T cells. So he gated the locate naive T cells by a surface marker, and then applied antibody linker, which is activate naive T cells. You can see the calcium influx occurs and returns to the ground state. And then they applied ionomycin, just acting as a control. So here it shows you a beautiful calcium influx plot. If you want to do a calcium influx, come to talk to us. And there's a certain, parameters on the software we can help you to set up. So usually you won't have this ratio exist. You won't have this ratio automatically you can find on the software, but you can add in this ratio onto your parameter. So we can also do a FRET experiment. And Simona from Chen's lab used the FRET to detect the membrane fusion. So she used this DIO-DII FRET test. So the DIO emits at green, the DII emits at red. So if you label the two cell population with either DIO and DII and incubate them together, so when the membrane fusion happens, you can see this population. So the signal from the green actually starting shift to the red. Not all the efficiency looks not that high, but in reality, as far as I understand, it never happens 100%. So you not only can use DIO, DII, there's other FRET pairs you can use. Some fluorescent protein you can use as well. So come to talk to us if you want to do a FRET experiment to detect whatever the molecule to molecules inside of your cells that will come in together very close within 10 nanometers of the range. We are talking about measuring the mammalian cells, also archaea cells. Flow cytometer can also measure bacteria. So bacteria here we measure E. coli. The size is around 1 micron. So to set up the parameters very important in order to exclude the background noises. Usually you will filter your buffer that dissolve, so suspend your bacteria cells. So cleaner, basically cleaner is better. So here it shows you the background noise coming from the buffer. And here shows your bacterial cell population here. So once we locate where the true bacterial cells are, we can put a gate around it and analyze further. Here, Rongzhen has putting a plasmid containing an antibody-resistant gene tagged with protein into the E. coli cells. So the GIP positive population, actually, that's what we selected for him. So the last but not least, the affinity selections. So I'm not coming across with affinity selections very, very often. So this is done a few years ago by a former member of LMB, LMB members. So basically, they established a system using DT40 chicken cell lines to select high binders to a therapeutic proteins. So they start loading the DT40, create a mini library of DT40 expressing this fragment, single chain variable fragments on the cell surface of the DT40. So they are libraries, they are small repertoire to start with. And these DT40 cells undergo intense diversifying of their genomes. So they incubate this library of DT40 cells with your target protein and select high binders. So after several rounds of selections, they're using magnetic beads first and flow cytometry later. So you can see for human interferon gamma, and they find high binders. So these binders, high binders, are further validated by ELISA to work out the kinetics. So this is the published paper if you're interested. Okay, so there's finally, and there's some reference for flow cytometry if you are interested. I'm sure you'll find lots of resources online. There are some of them if you're interested to look at it, or you come and talk to us. We can train you on our flow cytometers, especially analyzers. When we train you, we'll talk you through the basic concept of what is flow cytometer and what actually you're looking at. If you need help, then please contact us. So again, come and talk to us if you want to have a training or you want to store some cells, experimental design, anything related to cytometer, or you just basically want to have a look at our instrument. That's all. Thank you very much.